Look closely at this structure. Its geometric precision and spiked surface are highly evolved to find a host cell, break inside, and take over. For over a century, modern medicine has operated on a clear dividing line. On one side are humans, on the other, microbes. In this long-standing conflict, viruses have been cast entirely as the enemy. The evidence for this hostility is extensive. The 1918 influenza pandemic filled emergency wards with the dying, leaving tens of millions dead in its wake. Decades later, the poliovirus paralyzed thousands, filling hospital rooms with rows of iron lungs just to keep infected children breathing. In more recent decades, pathogens like HIV, Ebola, and SARS-CoV-2 have continued this devastating pattern. Through every era of recorded history, these biological agents have proven to be exceptionally efficient killers. In the 1940s, the mass production of chemical antibiotics like penicillin seemed to tip the scales. These broad-spectrum treatments were so effective, they gave Western medicine a profound sense of security. But that security was temporary. Bacteria have continually mutated, evolving defenses against our chemical weapons. We are now facing a severe crisis of antimicrobial resistance. This chart illustrates the trajectory we're on. While the blue line shows antibiotic efficacy plummeting, the red line tracks projected deaths from untreatable superbugs, skyrocketing toward 10 million people annually by the year 2050. At the same time, traditional small molecule drugs have reached a limit in their ability to fight complex solid tumor cancers without inflicting severe toxicity on the patient. With our chemical arsenal failing against both superbugs and rising cancer rates, researchers are turning toward a radically different biological solution. To find the solution, scientists had to stop viewing viruses as malicious entities and start treating them as highly evolved molecular machines. This begins with a mechanism called viral tropism. The surface of a virus is covered in protein ligands, molecular sensors that search for one specific cellular receptor. When the virus encounters a cell with a matching molecular signature, it docks with near-perfect precision. Understanding this docking mechanism allows for vectorization. Scientists surgically remove the disease-causing genetic sequence and replace that lethal payload with a therapeutic strand of DNA. The tough exterior protein shell, the capsid, remains entirely intact. It acts as a microscopic syringe, engineered by nature to penetrate cell walls. By controlling its target through tropism and replacing its core, a lethal pathogen becomes a delivery vehicle, engineered to ignore healthy tissue entirely. This brings us to bacteriophages, a specialized class of viruses first observed over a century ago by Felix de Herelle. Unlike human viruses, phages hunt only bacteria. When a patient with a drug-resistant MRSA infection is treated, the phage acts like a microscopic lunar lander. It touches down on the bacterial cell wall and injects its DNA straight into the host. This triggers the lytic cycle. The phage DNA hijacks the bacterium's own ribosomes, forcing it to produce hundreds of new viral particles until the cell wall degrades and the bacterium physically explodes. Because these viruses evolved with a strict tropism for bacterial receptors, they cannot attach to human cells. They pass through the patient harmlessly. In the face of mounting antimicrobial resistance, these bacteria eaters offer a scalable, self-replicating alternative to failing antibiotics.
In the realm of oncology, a similar strategy is taking hold. Cancer cells routinely survive by mimicking healthy tissue to evade the immune system. Oncolytic viruses strip away that camouflage. Researchers at Duke University spent decades modifying the poliovirus. They deleted the genetic sequence that causes paralysis in normal neurons, taking advantage of polio's natural attraction to a specific receptor vastly overexpressed on glioblastoma brain tumors. When this modified polio infects the cancer cells and causes them to burst open, it releases tumor antigens into the bloodstream. This massive cellular explosion trains the patient's own immune system to hunt down the rest of the cancer. Even the machinery of HIV is being utilized. As a retrovirus, HIV has the unique ability to permanently integrate its DNA into the human genome. Scientists extract a leukemia patient's T cells and use a deeply modified, non-pathogenic HIV vector to splice in a new genetic blueprint. This blueprint creates a chimeric antigen receptor, arming the T cells to identify and destroy leukemia cells. This table of clinical trial data demonstrates the reality of these treatments. Looking at the lentiviral vector row for the gene therapy drug Kymriah, trials achieved an 83% overall remission rate in pediatric leukemia patients. We have taken the very mechanisms that made polio and HIV so destructive and redirected them to clear terminal illnesses from the body. Despite these clinical successes, replacing traditional medicine with viral therapies involves severe logistical and biological obstacles. The human immune system is exceptionally good at destroying viruses. If a patient receives a therapeutic phage or oncolytic virus, their body will quickly generate neutralizing antibodies, potentially wiping out a second dose before it reaches the target. If the immune response is too aggressive, it can trigger cytokine release syndrome, a systemic, life-threatening inflammation cascade. Furthermore, cultivating pharmaceutical-grade living viruses without contamination requires massive, sophisticated bioreactors, making scaling up far more expensive than mass-producing chemical pills. Releasing self-replicating living medicine into the bloodstream requires a delicate balance between triggering an effective cure and inducing systemic toxicity. To overcome these limitations, current research is moving past modifying wild viruses and building entirely new ones, entering the era of the synthetic virome. Scientists are now designing completely bespoke viruses on computers. These smart phages are programmed with strict biological logic gates, ensuring they only activate when multiple specific disease biomarkers are present. By encoding these synthetic phages with CRISPR arrays, researchers can deploy them to precisely snip out the specific genes responsible for antibiotic resistance, forcing superbugs to become vulnerable to conventional drugs again. With rapid DNA sequencing, AI platforms will soon be able to analyze a patient's exact multi-drug-resistant infection and synthesize a custom-printed viral cocktail in a matter of days. Ultimately, by mastering and reprogramming the genetic code of the virus, we have transformed a biological threat into a programmable asset. The machines responsible for history's worst plagues are being rebuilt into the precise foundation of a healthier future for humanity.